Hello class 10. In my last video, I have uploaded the cell division chapter up to prophase. I hope you remember that I have talked about mitosis and meiosis. And I was teaching you the mitosis part, the first phase of the mitosis. Now today, I am starting with the second phase of the mitosis. It is the metaphase. What is this metaphase? The chromosomes arrange on the equatorial plane. Each chromosome gets attached to a spindle fiber by its centromere. Now students, I hope you know which one is the equatorial plane. I hope you know what is an equator in your geography. Here also, when we are talking about the cell, equatorial plane means the center part of the cell, the central plane of the cell, okay? And spindle fibers are those structures we have already seen in the prophase, that the chromosomes are clinging to that structure. These are the microtubular structures which are helping the chromosomes to be attached with it. So the main characteristic of metaphase is the chromosomes will arrange themselves along the equatorial plane attached to the spindle fiber by its centromere. Centromere is the part where the sister chromatids are joined to each other. Right. Now we'll see the diagram. See, I am showing you the metaphase stage of the animal cell. There is no early metaphase or late metaphase. Only one stage is there, that is only one metaphase. And I'm showing you, this is the animal and the adjacent one is the plant cell. Adjacent one, the rectangular one, is the plant cell. Okay. So we can very easily see over here, we can observe here that the chromosomes are lying along the equatorial plane of the cell, right? Okay. Now anaphase, the next phase. The centromere attaching the two chromatids divides and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. The drawing action is performed by the contraction of the spindle fibers. Now this again I'll explain with the diagram. Let us see the diagram first. Here again we will divide the anaphase into two stages, early anaphase and late anaphase. Now you see the difference. What's happening in the early anaphase? The sister chromatids are separating from each other from the centromeric region and is moving towards the opposite poles. And in the late anaphase, as we can see, very important point, that in animal cell we can see a furrow in the cytoplasmic portion. I hope you can see the furrow, students. Now this furrow is occurring only in case of animal cell, and in the plant cell there are no such changes that we can see. Only we are seeing that the chromatids, sister chromatids, are further contracted towards the opposite poles. They are moving further towards the opposite poles. So this is the early anaphase and the late anaphase, right? Last phase is the telophase. Each chromatid or the daughter chromosome lengthens, becomes thinner and turns into a network of chromatin threads. Nuclear membrane reappears, which was actually disappeared in the prophase. I hope you remember. And nucleolus also reappears in each daughter nucleus, which again disappeared during the prophase. All the contents of the cytoplasm, including the mitochondria and chloroplast in plant cells, are randomly distributed in the daughter cells during karyokinesis. Now, what is this karyokinesis? All the nuclear changes that occur during cell division are collectively termed as karyokinesis. And what is cytokinesis? Karyokinesis is followed by the division of cytoplasm, that is cytokinesis. From the name itself we can understand. Now let's see the diagram of telophase. As you can see, in the animal cell the furrow is very prominent, and in plant cell, as we can see, a cell plate has uh, appeared. The name of that cell plate, which has appeared from the between of the cell, that is the phragmoplast, right? And ultimately, by the cytokinesis process, as you can see in both the animal and plant cells, the cytoplasm has divided into two parts, into two identical daughter cells. At the end of the telophase, a furrow appears in the cell membrane in the middle, which deepens and finally splits the cytoplasm into two, thus producing two new cells. Now, difference in mitosis in animal and plant cell. In plant cells, the first difference is there is no centrosome, centrioles, asters, and are not formed, but spindle formation still occurs, which is formed by the microtubules. In telophase of plant cells, the cytoplasm does not constrict, furrow is not formed. Instead, a cell plate is laid down in the cytoplasm at the equatorial plane. It grows from the center to the periphery. Periphery means, the boundary, thus dividing the original cell into two daughter cells. Now, what is the significance of mitosis? Very, very important for your boards. Growth or increase in the body size, repair of damaged and wounded tissues, replacement of the old and dead cells, asexual reproduction in the unicellular organisms such as amoeba, yeast, etc. And last but not the least, very, very important point, maintains same chromosome number in daughter cells. Because if suppose the parent cell is carrying 23 chromosomes, then the daughter cells will also carry 23 each. Both of them will carry the same number of chromosome. Clear? Okay, now students, one thing I have to say in between that, the diagram which we have seen just now, very, very important both for animal and plant cells. Prophase, metaphase, early anaphase, late anaphase, early prophase, late prophase, then interphase, then anaphase, then telophase. Every phase you have to practice properly, because I'm telling you again and again, these questions are very common in your ICSE board exam, and definitely you will get question from this part of the chapter. So diagrams you must practice. Maybe you will get the diagram and you have to label it, or maybe they will give you for drawing the diagram. So in both the ways you have to practice. So practice it properly. Clear? Now we have seen a diagram of interphase while we were going through the various phases of mitosis. Now we have to know what is cell cycle, because interphase is a very important part of the mitosis. The new cells at the end of mitosis are relatively small with a full-size nucleus but relatively little cytoplasm. Now they will enter the interphase, during which they prepare for the next cell division and grow to the same size as their mother cell. So what is the utility of interphase? It actually prepares the cell for the upcoming mitosis, and also it helps the cell to grow in the same size as that of the parental cell. Now it has got three phases. Before we go through it, see the diagram. This diagram, students, is also very, very important. Please practice it properly. And we can see here the first growth phase, synthesis phase, second growth phase. We have named them as G1, S, G2. And from the diagram, we can see that G1 is the biggest or the longest phase. RNA and proteins are synthesized, the volume of cytoplasm increases, mitochondria and chloroplasts divide, these two organelles have their own DNA. In late G1 phase, all cells must follow one of the two paths. What are those? They may either withdraw from the cell cycle and enter a resting phase, which we denote as capital R, or start preparing for the next division by entering the next synthesis phase, that is capital S. Synthesis phase, more DNA is synthesized, the chromosomes are duplicated. And second growth phase, or G2, this is a shorter growth phase in which RNA and proteins necessary for cell division continue to be synthesized. Now the cell is ready to start next cell division, and thus the cell cycle goes on. So these are the three phases. You have to learn all the characteristics of all the three phases along with the diagram. Clear? Now one question they have given: can the cell cycle go on endlessly? What do you think, students? The answer is actually no. Why? Because if it cannot stop at a moment, at a point, then it will go on the cell cycle, and it will lead to a very, very rapid cell division process, which will lead to excessive cell growth, which will ultimately lead to tumors. So definitely that means it is very cancerous. So definitely cell cycle cannot go on endlessly. Clear? Okay, students, now meiosis is not there in your syllabus. As you know that you have to know that meiosis is a reductional division because it makes the chromosome number half, and also it is only it only takes place in the reproductive cells or gametes, right? So it's not there in your syllabus. This much is important. And one thing you have to know, that pairing and separation, when the pairing of the homologous chromosomes occurs, we call them as synapses. Also we call them as synapses. And chiasmata formation means crossing of homologous chromosomes chromatids. During pairing, some chromatids of homologous chromosomes break and rejoin at points of intersection. That is called chiasmata. So chiasma formation, it is there in your book also. You can see the diagram from there, and chiasmata formation diagram is also very important. Go through your book properly. And chiasmata formation is a very important part of especially meiosis, because chiasmata formation is the characteristic of meiosis which actually leads to the shuffling of the genes between the two parents, and then the offsprings, we get the characters of both the parents from the both the parents' genes. So this chiasmata formation is important for the shuffling of genes. So that is why it is a very important phenomenon in the meiosis cell division. What is the significance of meiosis? Chromosome number is halved in gametes so that on fertilization the normal number is restored. It provides for mixing up of genes. The maternal and paternal chromosomes get mixed up, and while these are separating, the chromatid material very often gets exchanged between the two members of a homologous pair, resulting in genetic recombination. Okay, the students, this is the diagram which I was talking about, chiasmata formation. Very, very important. And as you can see, they have shown with two different colors. When you are giving it in your exam, you have to also give it in two different colors to show the shuffling of the genes between the homologous chromosomes. Clear? Okay, students, so today up to this. Please go through the chapter. If you have any problem, come up with that in the comment box. And next day, I'll go through the second part of this chapter. Okay? Thank you.